It is located in cranium. It is divided into three parts: forebrain, midbrain, and hindbrain. Brain is comparatively short and broad, rounded in form, and whitish in color. It is completely fills the cranial cavity of skull. It is covered by two protective meanings that is membranes, an outer dura mater of connective tissue, and an inner arachnoid, which is rich in blood supply. Narrow space between two membranes contains cerebrospinal fluid. Brain has usual three principal divisions: forebrain, midbrain, and hindbrain. Forebrain consists of olfactory lobe. Here is olfactory lobe. This is cerebral hemisphere. Cerebral hemisphere. And diencephalon, which is not visible dorsally. Means first principal division, it is called forebrain, consists of olfactory lobe, cerebral hemisphere, and diencephalon. Olfactory lobe, which is first part of brain. Here is olfactory lobe. They are conical, very small in correlation with poorly developed sense of smell. Bird flying high above the ground, the bird cannot depend upon sense of smell. And hence olfactory lobes are poorly developed. They are conical and very small. Second division, cerebral hemisphere. There are two pyriform cerebral hemisphere are very large and convex, and two cerebral hemispheres separated by deep sagittal fissure. So this is sagittal fissure. They are relatively smooth and devoid of fissure. Cerebral hemisphere overhangs posteriorly, concealing the dorsal surface of the diencephalon completely. At the posterior end of deep sagittal fissure means here. At the posterior end of deep sagittal fissure, separating two cerebral hemispheres. Here two cerebral hemispheres separates. Here lies single, delicate, small, median, pineal body. On ventral surface of diencephalon, which is mostly visible, two optic nerve. The two optic nerve cross with each other, forming optic chiasma. Behind chiasma, project the median process. It is called infundibulum, which bearing a large hypophysis or pituitary body. Second principal division, that is midbrain. Midbrain consists of optic lobe and crura cerebri. Here is optic lobe. There are two rounded optic lobes. On the midbrain are conspicuously in keeping the keen sight of birds. The optic lobe becomes lateral because due to backward extension of cerebral hemisphere, and hence optic lobes are situated laterally. The two optic lobes are connected together by transverse optic commissure. Midbrain consists of optic lobe and crura cerebri. Then third principal division, that is hindbrain. Here is cerebellum and here is medulla oblongata. Cerebellum and medulla oblongata together constitutes hindbrain. Cerebellum is well developed and reached transversely. It is relatively larger. It is divided into a large median lobe, it is called vermis, and two small conical lateral lobes called floculi. Vermis, which is central, lateral lobe called floculi. Vermis and floculi together constitutes cerebellum. Cerebellum is large, much convoluted, that indicates the sense of equilibrium and great power of muscular coordination. Now here is medulla oblongata. Medulla oblongata concealed beneath the cerebellum, descend almost vertically from it to join the spinal cord. It is broader in front, taper behind to merge into spinal cord. It is triangular and curved part that continues behind as a spinal cord. Now that's about structure of brain of fowl. Then about functions. Olfactory lobes are concerned with sense of smell. Cerebral hemisphere concerned with voluntary movements, conditioned reflexes, instinctive behavior, intelligence, will, memory, emotions, etc. Diencephalon relay impulse to cerebral hemisphere. It acts as a integrating center of autonomic system and perception of extreme cold, heat, pain, and etc. Optic lobe concerned with sense of sight. Crura cerebri help in conduction of sensory impulses. Cerebellum help in voluntary movement and balance or equilibrium and muscular coordination. Medulla oblongata involves in involuntary movements like circulation, respiration, and heartbeat. So that's about structure of brain of fowl. Then dissection of fowl head so as to expose brain means a procedure for fowl brain. For dissection of brain of fowl, head is preserved in formalin. The head which is preserved in formalin is used for dissection purpose. Hold the head of fowl with dorsal side up. Take a longitudinal cut of skin up to tip of upper jaw. Cut the skin forward up to the snout from mid-dorsal line. Hold the cut portion of skin between fingers and stretch it outwards. Use scalpel to remove muscles. After taking cut, pull the skin on lateral side. Hold the head with one hand and pull the skin by other hand. Remove all the muscles with the help of forceps and expose the skull. With the help of forceps, pierce the cranium and remove cranium from all sides. That is anterior, posterior, lateral, dorsal. Remove pieces of cranium with the help of forceps. cerebral hemisphere, olfactory lobes, cerebellum are seen. Remove muscles from posterior part behind the medulla oblongata. Cut central part and sides with the help of scissors very superficially. Remove the pieces with the help of forceps. The spinal cord is exposed. Hold the spinal cord little just distance behind the middle oblongata with the help of forceps and pull it gently. Hold the spinal cord upward with fingers and slowly detach it from lower side with the help of arrowhead. Detach it completely up to cerebral hemisphere. Cut the olfactory nerve in front of olfactory lobes. Turn the head upside down. Observe optic nerves. Cut these nerves with the help of scissors and detach the brain completely from the skull by cutting all other connections. Keep the detached brain in watch glass containing water. Observe the various parts of brain along dorsal and ventral side. Black pepper can be kept below the brain or watch glass. Thank you for patient listening.